everybody. I'm Jayatri, Chief Bioscientist here at the Franklin Institute. And thanks for joining me for another conversation about the science behind the ongoing COVID-19 pandemic. It's been a week for COVID in the news, with President Trump's diagnosis and the spread of the disease throughout many of the White House staff. And so this is obviously a really important aspect that the national news is covering. And where I thought that it might be useful for us to address that timely aspect of the situation today was to talk about the medications that President Trump's been given. Because I think the description of how each of those medicines work kind of gives a really good overview of where we are in the big picture of the treatments that we've developed for COVID-19 so far. So I want to start there just to address some of these issues that are in the news. And then after that, I want to switch to this timeline for a potential vaccine being developed. Obviously, this is a critical development in terms of how we can project the course of the disease and the pandemic as we move forward. And there have been a lot of questions on how we determine when the vaccine is safe and effective and the statistics that we use to make those determinations. Now that's at the intersection of biology and math where I really like to geek out. We won't get too technical, but hopefully after our conversation today, you'll have a better sense of how scientists are looking at the data that are coming out of the vaccine trials, what to expect, and what the benchmarks are that are set in advance so we know when those results come out, what we'll be looking for. Okay, so let's start out talking about some of the drugs that have been in the news because they're involved in President Trump's treatment. The big three are remdesivir, a new cocktail of monoclonal antibodies produced by a company called Regeneron. And I want to make it clear that Regeneron is the name of the company, not the name of the drug. And dexamethasone. So let's talk about what these three drugs do, first of all. So remdesivir has been recognized by the FDA as being an appropriate antiviral treatment for COVID-19. Now, how this drug works is that it actually mimics one of the building blocks of the RNA that makes up the vaccine. So you administer the drug, it kind of interferes with the vaccine being able to make more copies of itself, and that shuts down the reproduction of the virus. I think I said vaccine before, I meant virus. So essentially remdesivir is a good drug to administer early on in the course of COVID-19 because it prevents the body from making more copies of the virus as the infection develops. Now, a second drug that President Trump's administered is this Regeneron monoclonal antibody cocktail. Now, that's a really long set of words for something that's essentially a synthetic version of convalescent plasma. So we've talked about convalescent plasma before. It's this idea that if you can take blood from patients who have recovered from COVID-19 and isolate the antibodies that their bodies produce, then you can take those antibodies, give it to somebody else who's been infected, and basically give their immune system a boost. Now, that's shown some promising results. It's in clinical trials right now. And the idea of creating this cocktail of monoclonal antibodies, monoclonal just means it's an antibody that recognizes a single target. So is that you can take the best antibodies produced either by the human body or in mice that have been injected with the COVID-19 virus. So you take the best antibodies and you isolate them, you put them in this specific cocktail, and you can administer them directly. So it's essentially a synthetic way of taking the best and most effective antibodies that are naturally produced and using that to, again, give the patient's immune system a boost. Now, this is very much experimental. There haven't been any peer-reviewed clinical trials on this to date. So far, there's been promising results that have been published in a press release that seem really exciting. But as I've said, it is very experimental. So, we won't really be able to tell from, its use to treat the president in terms of, what it might mean on larger populations. So that's still an open question. And then the third drug that's kind of in the big three that President Trump's gotten is dexamethasone. Now, this is more of a generic steroid drug that brings down inflammation in the body. So this drug is generally used at a different stage of infection than the other two. So the other two, whether you're trying to decrease the viral replication with remdesivir, or you're trying to boost the immune response with the antibodies, those are both to help the body fight off the infection at an early stage. What dexamethasone does is that it prevents inflammation when the body has progressed to a later stage of disease, where you're starting to see more infection in the lungs. So there's some questions here about exactly, what stage of infection President Trump is at. And I'm not a doctor, so I'm not going to comment on his particular case here. But that's also been shown to be effective in reducing that level of inflammation. So these three are kind of the most targeted drugs to help both fight the infection and to reduce the probability of the body overreacting with its immune system. Now, I wanted to mention the hydroxychloroquine because that's a drug that's been proposed as a possible treatment and is still coming up in the news. But there have been continued studies that show that hydroxychloroquine just doesn't have much of an effect on COVID-19, and so it's not considered to be part of the standard of care. Now, in the press releases issued by the president's doctor, there were a couple of other medicines as well, and vitamin supplements that were mentioned, and I just want to touch base on them because people may have questions about them. So one is vitamin D. This is a kind of an interesting one because there have been observations that people who have lower levels of vitamin D seem to have a higher risk of contracting COVID-19. Now, right now, all we know about this association is that it's just that. It seems to be correlated. And we don't know whether whether having low levels of vitamin D causes that higher risk or whether there's some other factor involved. And so vitamin D right now is, again, in clinical trials to try and study whether there's a direct effect or whether it's just this correlation. So we don't know for sure how vitamin D might help right now. Similarly, zinc is another mineral. It's used in chemical reactions throughout the body. So we don't, it's not a specific target that might be, related to the disease. Again, what we know is that in infectious diseases more broadly, not just COVID-19, that having low levels of zinc can put you at higher risk. But again, we don't know whether there's any specific effect of zinc at preventing or treating COVID-19. Again, there are clinical trials underway to start pulling this out. Then there are two other supplements that one was called famotidine. It's a generic name for what we might be more familiar with as over-the-counter Pepcid AC. You might be taking it for heartburn. Again, there are some very early observational data that suggests that maybe Pepcid has some anti-inflammatory effects that could help, but again, we don't really have solid evidence for a therapeutic effect yet. And similarly, melatonin. Again, there's some hypotheses and observations that melatonin can have an anti-inflammatory effect. Again, we still don't have solid evidence there yet. So all of those together, I think, help paint a good picture of where these different treatments are. We have some that are, that seem to be part of the standard of care based on the evidence that we have now. There are some, like this Regeneron antibody cocktail, that is promising, but we just don't have the evidence yet. And then there are some other vitamins and supplements that are so far still observational and need more study. And so, again, one thing that I want to remind everybody is that how scientists evaluate whether these drugs work is based on population level data. We have to look at large numbers of patients and do comparisons between people who are getting the drug and people who aren't getting the drug. And so for one patient, even though he's a very high profile patient, we won't really be able to draw any conclusions about the efficacy of any of these particular treatments. So I think that hopefully gives you a picture of how doctors are approaching his treatment and why he's getting some of the drugs that he is. Okay, feel free to pop some questions in the chat if you have anything that came up about that. But let's move on to talking about vaccine testing. So there are 3 vaccines that are sort of leading the pack right now in terms of where they are with phase 3 trials, which use a sample size of tens of thousands of people. I want to throw a caveat in here that these do not include, these 3 that I'm going to talk about don't include the vaccines that are being developed in Russia and China, because we don't have as much information about them, and so I can't really compare them in the same way that I want to walk you through the process that we know for these 3 vaccines. And the 3 vaccines that I want to summarize today are the Moderna vaccine, the AstraZeneca vaccine, and the Pfizer vaccine. Now, what's interesting about these, and I'm sure there many of the other companies who are working on vaccine development will follow this precedent, but these 3 vaccine companies actually release their entire detailed protocol for how they plan to test the efficacy of their vaccines with different milestones for both efficacy and safety. And this is kind of unprecedented. Most of the time, these kinds of protocols are confidential. That they're kept within the company, they're kept within the study group, so as not to necessarily reveal their methods. But one of the things that everybody recognizes is essential to the success of this vaccine is trust in the vaccine. How can we trust that this vaccine is safe? So, before we go there, let's pause for a question.
Keep hearing that vaccines are being fast-tracked. Does that mean it's more dangerous?
That's a great question, and that's exactly the question that I want to answer today. So, before we get into that question, what does it mean for safety? I want to talk about, what does it mean that these vaccines are being fast-tracked? So, I think the most relevant point to think about, how we've gotten from the sequence of the virus in January to, vaccine candidates being tested in tens of thousands of people in October, is that we've learned a lot about vaccines over the last few decades. That, I don't follow a lot of football, but I was talking to a friend who does. Being here in Philly and not being an Eagles fan, I feel a little bit of shame about not having a great football analogy at my fingertips. But my friend was talking about the fact that maybe it's analogous to starting vaccine development being in the red zone. That we are close to getting the touchdown because of where we're starting from. We're not starting from scratch with these vaccines. The vaccine development work that's gone into research and development of vaccines for previous viruses, whether we're talking about the original SARS virus or the Middle East respiratory virus or Ebola or HIV, all of this work kind of brings us to a starting point much, much farther along in the process than where we had been before. And so that is one big reason about of why we've been able to get to where we are as fast as we have. And we can drop a link in the chat to a previous conversation that we had about the science of some of these vaccines and how they work to follow up on how that previous work has led to the development of these particular vaccines. But to your second point, how can we be confident that these vaccines are safe and effective? And that's exactly why these companies are trying to be really transparent about the process by which they're evaluating them. So let me walk you through kind of a high-level process of how these vaccines are analyzed. So this is where the math starts to get important, because it all starts with predictions. So one thing that's critical to a good scientific experiment is to predict what you might see, predict what your null hypothesis might be. If you go back to your scientific methods class, that what am I trying to disprove, and then calculating the number of participants you need based on on several assumptions to be able to essentially disprove that the vaccine is not effective. So that's this double negative, right? You want to prove that a vaccine is effective, but the way that scientists think about it is that we have to disprove the negative. So we're trying to disprove that the vaccine is not effective. Okay, let's talk about what that means. So when these phase 3 trials start, before they ever even recruit a single participant, you need to predict how you're going to know whether your vaccine works. You have to predict whether you will be able to disprove that null hypothesis. And so the way that they predict the number of participants that they're going to need depends on a couple of assumptions. One is that they're going to assume a certain target vaccine efficacy. So a common one that all three of these companies are shooting for a target of 60% vaccine efficacy. What does that mean? That means when you compare people who got the vaccine to people who didn't get the vaccine, you'll see 60% fewer infections in the vaccinated group. That make sense? So again, you've got one group of people who get the vaccine, another group of people who don't get the vaccine, and if the vaccinated people get at least 60% fewer infections, then that tells you that your vaccine has 60% target efficacy, or vaccine efficacy. So that's a term that you're going to hear a lot, is this idea of efficacy. So that's something that the scientists kind of set what their goal is. The FDA, just for reference, has said that they will approve vaccines that are more than 50% vaccine efficacy. So they're trying to shoot for better than what the FDA standard is. Okay, then there are a couple of assumptions. And one important assumption is what the infection rate is in the community, where the participants in the trial are, live and work and play. So this is something that depends on where the vaccine trials are being conducted. So obviously, if you live in an area of the country where there's a high frequency of COVID-19 right now, you're more likely to get infected. And the vaccine companies are trying to do their trials in those parts of the country to try and increase the likelihood of seeing the effect of the vaccine sooner. If you live in a part of the country where there aren't many infections, then you're just not going to get your results as fast. So that's a pretty simple rationale there. And most of these companies are kind of estimating an infection rate of between 0.65 to 0.8% infection rate. So that's another calculation that affects what that prediction is going to be. And there are other assumptions there too. I'm simplifying down to the main ones right here. But the other key factor in predicting the number of participants you're going to need in your trial is how much statistical power you have. So what is the probability that you will be able to detect a significant effect? And most standard statistical analyses want to have a 90% chance of detecting that effect. You want to give yourself as good a chance as you can to find what you're looking for. So those are three key elements that go into estimating how many participants you're going to need. And we'll talk about why that number can change. Because essentially, if the vaccine efficacy or the infection rate changes, you might see the effect sooner or more strongly than you might expect based on your initial assumptions. Okay, so all of these companies have their predicted number of participants, so they start recruiting, and they all have a vaccine arm and a placebo arm. So the placebo group is not getting an active vaccine; they're getting basically a sham. Whereas the vaccine group is getting the approved the experimental vaccine, and all three of these companies are using a 2-dose vaccine, where you get 2 doses of the vaccine a few weeks apart. So they start recruiting people, and as they recruit, they're putting them into each of these groups equally. So they're kind of building up their participant pool in parallel between the vaccine arm and the placebo arm. And then they start looking for cases of COVID, right? When you're looking for how to analyze vaccine efficacy, you want to compare the number of infections that you see in the placebo group to the number of infections that you see in the vaccine group. And hopefully, if you see many fewer infections in the vaccine group, then you know your vaccine is working. And so this is where you can start to see the effect before you hit your total number of predicted participants. And so what these vaccine companies do as part of their protocol, again, this is before they ever start recruiting a single participant, is that they actually declare some interim analysis points. So I'm going to give you an example in a minute from the Pfizer study, because they have 4 different interim time points before they get to the final number of participants. This varies from study to study. The Moderna study has 2 interim time points. The AstraZeneca has 1. And these are kind of check-ins that when you get to a certain number of cases in your entire pool, you can do some early analyses to see where you are, right? Maybe, and if you're lucky, then maybe you see an effect right away. Depending on some of these variables and the assumptions and what's borne out in reality, maybe you don't see it right away and you have to keep going. But again, these kind of checkpoints are designed into the study. These aren't, they're not cherry-picked, and that's a very, very important thing to know. Because these are things that the review boards will be looking for, is that they're sticking to their original plan. So that's important for analyzing the efficacy of the vaccine, and then of course monitoring side effects to ensure safety. And again, I'll talk about the Pfizer example and how they're monitoring it, but this is the part that takes time. So efficacy is a fairly straightforward thing to measure, right? Either people are getting infected or they're not. The safety part is where you have to follow people over time, weeks, months, and so this means that you can't, just because you might see evidence of efficacy doesn't guarantee that it's safe until you have more of the participant pool monitored for a long period of time. Okay, so let me break down for you the Pfizer study. Because they have the most checkpoints, I think it's a good example. Before I do that, let's pause for a question.
We have multiple, actually.
Okay, great. Throw them at me.
Okay, so first is, what makes the previous work a good starting place for the vaccine of COVID?
Yeah, okay, so there are a couple of platforms or delivery methods that current vaccine efforts are building upon. And some of them are kind of tried and true. So for instance, we've referred a lot to the spike protein, for instance. So the spike protein on the coronavirus is a part of the virus that sticks out of that ball of virus. And it's the part that binds to the cells in our body and kind of gets the infection started. So you can isolate the gene for the spike protein, put it in another virus that doesn't make you sick, and essentially use that other virus to introduce the spike protein by itself in your body. And so that then helps your body recognize the spike protein, develop an immune response. But that delivery virus, which most often is something called an adenovirus, has been very well established as a vaccine delivery device. So that's an example of an established platform that's been well tested, and it just needs to be modified with this new part of a new virus to be able to be delivered. Another tried and true method is to just isolate part of the spike protein itself and deliver that as a vaccine. Now, one of the newer platforms that both Moderna and Pfizer are using is to actually use just part of the RNA, the gene sequence of the virus directly. And although that platform has never been approved in a vaccine, they have been working on this for several other viruses, including Ebola and MERS. And so even though it hasn't yet gotten to approval because those viruses kind of petered out before we needed to scale up vaccine development, we were already kind of halfway there. All right, next question.
Okay, so let's see here. What's the goal with having a vaccine? If you don't receive the vaccine and you get the virus, can you be treated with medication without being hospitalized?
That's a great question, because one of the puzzles of COVID-19 is the fact that it seems to have a whole range of severity when somebody gets infected. And we know that there are some risk factors, such as other conditions like lung, breathing conditions or heart disease that might predispose you to a more severe case. But we're still trying to figure out why some people get really, really sick and other people barely have any symptoms at all. And that variation is one of the biggest reasons of why a vaccine can make such a difference, right? If everybody just got kind of like a common cold and we could deal with it, right? It's fine. But the problem is that some people get really, really sick, and so having this whole menu of options of drugs, like we just talked about, that President Trump is being treated with, for example, is another toolbox of how to treat people once they've already been infected. But if we can have a vaccine that helps prevent people from getting infected in the first place, that's just better for everybody.
Okay, next is, should you get the vaccine if you've already had COVID?
Another great question. So we don't know is the answer. And actually, we can talk a little bit about that these vaccine testing protocols all have primary objectives, and the primary objective is whether it can prevent infection in people who have never been infected. But some of them also have secondary objectives. And I can't remember which trial it is, but one of them, the secondary objective is what happens in people who have already been infected. Because we don't have a good understanding of whether people can get reinfected. There are some individual cases that I've heard of where somebody has gotten COVID and then recovered and then gotten it again. And by sequencing the genome of the virus, scientists have been able to tell that they do seem to have multiple infections. But those cases seem to be kind of few and far between right now. So that's not the priority for the vaccine testing right now, but it's certainly a question that we still need to answer.
Okay, and then finally, heard that a trial was shut down because someone got really sick. Does that happen often in these kind of trials?
Yeah, that's a great question. And so that's referring to the AstraZeneca trial, which was shut down for a week or so because there was a participant who had a severe side effect to it. And that is not uncommon. And the fact that the trial was shut down is actually part of exactly how these trials work. So one thing we haven't talked about yet is that there are external review boards that monitor the data and the results coming out of these trials, so that they're being reviewed by an independent group of scientists who are not part of the company, so they don't have any stake in its success from a financial point of view. And so when a company finds an adverse side effect in its trial, it has to stop and then share the data with these independent review boards. And that gives them time to analyze the data and study that particular patient to see whether this side effect was likely to be due to the vaccine, or whether they might have had some other condition that resulted in this, or whether there's some interaction between the two. And just because there's one side effect in one person doesn't mean that the trial gets stopped unless something very obviously dangerous comes out. But it's just a pause to kind of wait, look into the data, think about why it happened, and assess what the risk might be to the other participants in the trial. So that trial has restarted again. And again, this is a fairly common thing that happens in these trials and is part of the process. That's a good opportunity to think about the fact that there are independent scientists who are reviewing all of the procedures and the data that are coming out of these trials. Okay, any other questions?
Nope.
Okay, let's dig into the Pfizer trial, and I give this as an example because they had this lovely table in their protocol that I think helps us understand what these interim milestones that they're looking at are. So again, based on those assumptions that we talked about when they're predicting the initial number of participants, things like what do they expect the vaccine efficacy to be, what the local infection rate is, and what their statistical power is. All of these trials are looking at roughly 150 to 160 cases of vaccines, or cases of COVID, among the entire study group of tens of thousands of people, to be able to have, again, that statistical power to detect a difference. So what does that really mean in terms of actual numbers? And so this is, I think, a useful explanation to help understand this. And so for the Pfizer study, their final goal is to look at the data when they have 164 cases of COVID. Now that's 164 across both the vaccine group and the placebo group. And what they're going to be looking at is how the COVID cases are divided up between the vaccine group and the placebo group. But they have these earlier checkpoints that they're not going to wait until they see 164. They have 4 different interim checkpoints. The first one is when they see 32 cases, 62 cases, 92 cases, and 120 cases. So let's look at what they predict to see in, oops, I'm getting some direction that my board is not well positioned. Is that better?
No, I mean like move yourself over.
Oh, okay.
You're like,
how's that?
that's better.
All right. Thanks to my director here. Okay, so let's look at what they're looking at these different early time points. So when they hit 32 participants who have contracted COVID, no matter how many people they've enrolled in the study so far, they're looking at 32 cases of COVID, they're going to stop and look at the data. And if you were to see a successful outcome of the vaccine, you would expect to see 6 or fewer in the vaccine group and 26 or more in the placebo group. And again, based on their calculations in their assumptions, this distribution at just 32 cases is such an overwhelming difference that you can say with confidence that this vaccine is effective. On the flip side, if you see numbers like 15 COVID cases in the vaccine group and 17 COVID cases in the placebo group, then you kind of know that your vaccine isn't very effective, and at least so far. And they're going to keep going at this point, but it's not looking great. Okay, so they keep going with the trial, and the next milestone is when they hit 62 cases of COVID. Again, they're going to look at how that 62 breaks down between the two groups. If they see, 15 in the vaccine group and 47 in the placebo group, again, that's numbers that are successful. If you see 26 in the vaccine group and 36 in the placebo group, not so good. Again, next checkpoint is at 92 cases. If we've got 25 in the vaccine versus 67 in the placebo, that's great. If you see 35 in the vaccine versus 57 in the placebo group, that's not great. And if you see this trend over 3 different checkpoints, at that point they decide that it's just not worth going on. And so that's their milestone for saying, stop the study. Likewise, if you keep going at 120, you see 35 versus 85 cases in vaccine versus placebo, or in the final 164, if you see 53 versus 111, those are signs that the vaccine is having a good enough effect that, this is worth pushing on. Okay? So when you hear things like, we could know as early as October as to, whether the vaccine is effective, that is a best case scenario. So that is looking at, this first interim milestone and seeing this kind of result. However, that has a lot of assumptions. That's assuming that the vaccine is really effective. That you're looking at, not just 60% effective, but maybe 80% effective. And you're looking at maybe a higher rate of infection than you assumed. So that means people are getting infected faster, and so you're seeing the results faster. So when you see people talking about early results, they're not making that up, but it is a best case scenario that is unlikely. Okay, so I hope that just to kind of temper the expectations around that, these are predictions based on statistical calculations, but the assumptions that would result in these numbers are unlikely to be met. So hopefully that gives you a sense of how these trials are conducted, so that you do have these different, preliminary checkpoints before you get to the final that give you some indication of how your study is going. But again, I can't emphasize enough that the reason that that these checkpoints are valid are because they're calculated in advance. That they've already declared what when those checkpoints will happen and what is predicted, so that you can't just make them up as you go along. So hopefully that makes sense. Now, of course, the efficacy is just one part of the vaccine trial. The other part is the safety. As I said before, that's something that takes time. Just to give you an understanding of how a vaccine company might monitor that, they actually track side effects, for every patient. And Pfizer, for instance, classifies those side effects into what they call tier 1 side effects. So these are the side effects that they saw in the early phase 1 trials that they kind of expect. Things like soreness in the arm, or maybe a low-grade fever, kind of minor things that they expect. A second category is what they call tier 2 side effects that aren't already predicted, as you'd find in the tier 1, but they're seeing at least 1% of patients in a certain group of participants being affected, so that they're more common, and that's worth studying. And then there are, of course, the tier 3 effects that don't fall into either of those categories. They're not the minor ones that are expected. They're not ones that are common among others, but they are seeing them in patients and could potentially be severe. And again, all of those definitions are laid out in these protocols. So again, that's really important because everybody's on the same page about what counts as a side effect and what happens when a side effect or potential adverse event happens. There's a plan for that. So these protocols are, like 100 pages long, and they've gone through numerous rounds of review. There are ethical boards that review these protocols to make sure that patients who enroll the participants are getting the information they need about the risks and the benefits. There are external data monitoring boards who are looking at both the safety and efficacy numbers. Obviously, all of these are done in what we call a double-blind fashion, so that neither the patients nor the scientists who are working with them know whether any given patient is getting the vaccine or the placebo. And then, of course, the FDA has a separate review board as well, called the Center for Biologics Evaluation and Research. That is another layer of external review. So all of these different review processes are in place to make sure that the data are being analyzed correctly, that the appropriate safeguards are in place to protect the participants, and to make sure that the data that are being published are transparent and complete. And it's really important to know about the process, not just of how the trial is conducted, but also that there are these checks and balances to ensure the integrity of that process. So I hope that gives you a good understanding, at a high level, as to how this process works and how to interpret the news that comes out, and where you can take things with a grain of salt, and how to dig a little bit deeper when you hear you hear some of the preliminary results start coming out. You'll be able to know that, oh, okay, that's their interim analysis number 1 when they've got 32 cases. I know what that means. Let's pause for a question.
What type of timeline is this vaccine being treated without hospitalization, schools reopening, masks, and strict protocols in the community being lifted?
That's a great question. So when this vaccine, multiple vaccines are approved, what does it mean for a timeline of when life can get back to normal? And the answer is that we don't know. There are certainly efforts to scale up and manufacture enormous numbers of doses of vaccines, again, faster than we've ever done before, both through government efforts as well as private efforts. And so, again, that gives us a leg up. But ultimately, the effectiveness of the vaccine is going to depend on how quickly we can distribute them, who gets it first, if we can get the vaccine to the people who are at the highest risk, and ultimately how many people choose to get it. What we know from infectious disease epidemiology is that the more people that get it, the more we protect each other. And that's why it's really important for scientists to explain the process so that people have that trust in vaccines. And the more people that get it, the faster we'll get to a point where we're protected from the disease and we can start to get back to normal. Any other questions?
Nope.
All right. Well, thank you so much for joining me today. Like I said, we'll drop a comment in the chat about some of the other conversations that we've done about vaccines if you want to go back and get some more details on those. But thanks a lot for checking in. We'll be back as the news develops. Thanks a lot and stay safe.